This is Dr. Saad in front of you, and today my topic is the mutations in the transcription factors. As we were discussing the first hallmark of the cancer, that was the self-sufficiency in growth. So now we are moving on towards the its last part, that is the transcription factor. As I told you that we have the growth factor, then it activates the RAS, and RAS further activates the two pathways, that is the RAF and phosphoinositol 3 kinase. Then RAF further activate MAPK, which further move into the nucleus, and then it moves towards the transcriptional factors. Then I told you here that it activates the AKT pathway, where it activates the further you can say its substrates, that are the mTOR, FOXO and the BAD. So these all part we have studied, mutation in RAS, mutations in these all we have studied one by one. Now the last part that was remaining is the transcriptional factors. So now we are moving on to the transcription factor. What happens that normally that the all transduction signal pathways, they finally move into the nucleus, and what happens in the nucleus? Obviously there will be the gene expression in the nucleus. Clear? So then what happens that the transcription occur. So we have the certain transcriptional factor genes, that means that their products that are the transcriptional factors. And what are those genes? For example, the most important gene is the MYC gene. This is a MYC gene, that is a normal proto-oncogene. We have studied here MYC, that is a normal proto-oncogene. If the mutation occur, it becomes oncogene. So MYC normally it is a normal gene, and what happens, it carry out the cell cycle growth. Clear? It carries out the cell cycle growth. Now you can see this is a protein, and its product is a transcriptional factor which carrying out the cell cycle progression. Now important point here that the mutation occur in the MYC. How the mutation occur? Basically what happens that on the chromosome we have, means every gene it is regulated, it is well regulated. So MYC is also regulated by certain other genes. Clear? You can say that there is a negative regulator for this MYC. So there may be a loss of function mutation in those negative regulator, and which results in the gain of function of the MYC, which results in the growth and cell cycle progression. Clear? So in this way there will be the mutation in the MYC, or you can say the mutation in the negative regulator of the MYC. Clear? Now basically this MYC, this gene is mutated in the fastest growing human cancer, that is the Burkitt's lymphoma. Clear? You have to remember this. It is mutated in the Burkitt's lymphoma. This is very important point. And one thing, that this is the main, you can say main transcriptional factor, or you can say master transcriptional factor is the MYC. Means the most important main transcriptional factor is the MYC, and it is the most common transcriptional factor that is mutated in the human tumors. Clear? We also have other transcriptional factors, means we have the MYB we also have, and others we also have, but the main focus is the MYC that we are studying in the detail. So this MYC, it is not only means mutated in the Burkitt's lymphoma, but it is also mutated in other, like the lung carcinoma, colon carcinoma, breast carcinoma. So we have two types, you can say NMYC, according to the means cancer in which they are mutated. N for the neuroblastoma. Then we also have the LMYC. This L stands for the lung carcinoma or lung cancer. Clear? So according to the name of the cancer, we also have these NMYC and the LMYC, means they are mutated in these cancers. Clear? So this is the MYC. Then we also have studied about the normal pathway. In the normal pathway we studied about the cyclins and the cyclin dependent kinases. So what are the cyclins? And this is all about the MYC. Clear? The mutation in the MYC and where it is mutated is the main transcriptional factor, master transcriptional factor. It is also mutated in these cancers, and many other cancers is also mutated. Now we are moving on towards the cyclin dependent kinases and the cyclins. Basically we have the cyclins and cyclin dependent kinases. Clear? Now what happens that they also carry out the growth cell cycle progression. Obviously they are the part of the normal growth pathway, so also they carry out the growth. But how they carry out the growth? Basically what happens that these cyclins and cyclin dependent kinases, they forms a complex. The complex is formed between these two, and this complex, it basically phosphorylates the proteins. It phosphorylates proteins, and these proteins then they carry out, means they are activated, and now they are carrying out the growth and cell cycle progression occur. So in this way there is a normal pathway. And normally one more thing is present here, means this pathway, I already told you that all genes, they are negatively regulated. So this pathway is also negatively regulated by cyclin dependent kinases inhibitors, means those that inhibit the cyclin dependent kinases. So they normally inhibit these so that the cell cycle may not progress very fast or may not over proliferate. The cell cycle or the cell may not over proliferate. So they are the negative regulators that are normally present. What happens that now if the mutation is occurring in these, what happens that there will be the gain of function mutation in this. In the cyclin dependent kinases, there will be gain of function mutation in these. Clear? And one thing that there will be loss of function mutation here in the cyclin dependent kinases inhibitor, so the cancer may develop. Clear? Means there will be loss of function, means there will be no function of these cyclin dependent kinase inhibitor, means they will not inhibit these cyclin dependent kinases. When they are not inhibited, the cell cycle will progress. Clear? Will more progress when it is means move from the negative regulation. And if there will be gain of function mutation in this genes in the cyclin dependent kinase, obviously the cell cycle will proliferate and progress more and more fast. Clear? So this is all about the first hallmark of the cancer, that was the self-sufficiency in growth. Now in the next lecture we will be starting our next hallmark, that is insensitivity to the growth inhibitory signals. So if you have any question, any query about the first hallmark of the cancer, you can ask in the comment section. And thank you so much. Allah Hafiz.